Hi, everyone. Welcome to the first event in our new MetroFlow corporate member launch seminar series. It's 12 o'clock. We're going to wait just a couple of minutes. We have about 35 people on the seminar thus far. So we're going to wait another minute or two. But thank you, everyone, for joining us today. We're going to be using the Q&A button that you find at the bottom of your screen for questions. So let's go ahead and test out the functionality of that now. So if you could, go ahead and type in as a question where you are viewing us from today. What part of the world are you in? Wow. Hi from Mainz, Germany. Wonderful. Thank you. Boston, Massachusetts. We have 2 people from Maryland. Jason is still on Long Island. Great. Florida, Manhattan, Fort Myers, Florida, New Jersey. That's where I'm at today. New York, Jersey City, Tampa, Florida. A lot of people in Florida. Augusta, Georgia. Fabulous. Well, I think our attendee from Germany definitely wins the distance award for today. So thank you for joining us. Okay, let's go ahead and get started. So again, I will say welcome to the first event in our MetroFlow corporate member launch seminar series. I am Christina DeCoste, Director of the Flow Cytometry Resource Facility at Princeton University and President of MetroFlow, the New York, New Jersey Flow Cytometry Users Group. Since we are unfortunately not able to meet in person during this time, the 15 members of the MetroFlow Steering Committee have created this launch seminar series as a vehicle for our corporate members to reach our MetroFlow audience and others via this virtual platform. This session is the first of several to follow throughout the remainder of the summer. And as a disclaimer, MetroFlow remains neutral and in no way endorses any one vendor or their offerings over another. All corporate members have the opportunity to submit a proposal to present in this launch seminar series, and the purpose of these events is to showcase the innovations in the field that we can all take advantage of to better support our scientific and research endeavors. We hope that all of you will be able to take what you learn from this seminar series and those that follow back to your labs, and that the information will be helpful to all who are watching today and in future seminars. For some housekeeping on the Zoom platform, select View option, Fit to Window, found on the top of your screen for best viewing. And please use the Q&A button at the bottom of your screen to submit questions, which will be read to the speaker at the end of the presentation. Please use the chat function only to report any technical difficulties. And before we get started, we'd like to thank today's speakers, as well as you, our MetroFlow members and extended audience, for your time and participation. And with that, I'd like to introduce our first speaker, Angie Goldfein, the Scientific Director at Propel Labs.
Hi, everybody. Thank you so much, Tina, and the rest of the MetroFlow committee. We really appreciate this opportunity to present some more technical detail on the Bigfoot Cell Sorter. We're going to be focusing today on high throughput, but in case you missed our launch last week, I'm going to give you a quick overview of some of the high-level features of this brand new cell sorter. The Bigfoot is a 9-laser, 60-parameter cell sorter. The video. Bigfoot, sorry, I'm making sure we had video. 9-laser, 60-parameter cell sorter with spectral capabilities. So we can do both spectral analysis and sorting. It's a 6-way sorter with accuracy for up to 1536 well plates. Just a brief overview of some of the other features of the system. You'll notice it has an enclosed biosafety hood. This is a Class 2 containment hood built into the system. We started our design with this, so it's really focused on safety for you and your sample from the beginning. You'll notice the adjustable sash that can move up and down, and this maintains an even opening no matter what the use is, always keeping you safe and keeping constant airflow. Inside to the right, you'll see the sample loader. We actually have a 6-position sample loader that can use tubes from 1.5, 5, or 15 ml. We even can sense the type of tubes you have in there to adjust the pressure accordingly. It has built-in agitation and temperature control, and also a bead station. So we have onboard QC beads so that in the morning you can start up the system and have it do its quality control before you even walk in the lab. The QC is a single peak for alignment, a double peak for spatial separation, and then a peak for drop delay, which we calculate onboard the system via a dedicated port on the waste catcher. Moving up, we have a user changeable sample line. So from here, you can change the sample line between samples if you're doing sticky cells or just want to swap it after use. Next to that is our nozzle. Now we have the nozzle on a 5-axis mount, and we can auto align the nozzle to the laser beams to really optimize alignment on a daily basis. You notice when I press that door open, it comes out to you. So it's in this position for easy nozzle swap and changing for different tips or unclogging. We offer tips from 50 to 150 microns. Below that, you'll notice the sort output chamber. Now, of course, this has its own dedicated aerosol management system as required by some of the NIH guidelines. And we have a built-in way in the software to help you test that regularly in your laboratory. In addition to the temperature control and the plate sorting, which you're going to hear about a lot later, I wanted to talk a little bit about the other tube sorting options. This is the sort output module. We have 3 rows of tubes, so you can do 3 rows of 5 ml tubes, you can do 1.5s, or a mix of 15 and 50. This allows for virtual 18-way sorting that you can set up in advance. Less time that you're reaching in and out of the hood and swapping out samples. These easily pop in and out so that you can swap them. And for the 1.5s, we even have a place for the caps so you don't have to clip those caps off anymore. Lots of flexibility there. I'm going to let Nate and Mike go into more of the sort detail though. You'll notice there's a lot of space inside the hood here. We have a built-in vortex, so you can bring your tube rack in, vortex your samples, throw the cap away in the included biosafety bag, and then run. We're limiting your in and out of the system and trying to streamline your workflow as much as possible. A few other things. Down here, we have onboard bulk fluids. We actually have dual DI and waste tanks. You can trade them at any time. Even if you're in the middle of a sort, you can swap those tanks and keep on going. You don't have to stop anymore and refill tanks and then recalculate your drop delay to finish your sort. We have a bottle of additive as well, which we dilute on board to create your sheath solution. 2 cleaner bottles, a decon and a cleaner. Those can be used for shutdown or between samples to keep your system clean. And those can be programmed to be used just the way you want them. On this other side, you'll notice we have our detection system. Now, I did mention up to 60 detectors. We do allow you to change the filters though, the bandpass filters and the last dichroics right before the detector. So as new dyes become available or as your lab grows, you can change those. This huge array though gives us that capability to do spectral sorting and analysis. So you can do traditional compensation or spectral, totally up to you. There's a lot of things in our workflow that make spectral really easy to use. A few other features of the system. We have built-in storage. Keep all your parts in one place, your sample adapters. We even have a 10x adapter so you can sort directly into 10x plate. Just nice to keep all your extra tools, nozzle tips, sample lines, all in one place, right at the tip of your fingers. Over here, we have an adjustable monitor and computer. So you can adjust this keyboard and monitor to any height you like. This is really valuable so that when you're across the room, you can look at it and see, is your sort done? Are your bulk fluids? Do they need your attention? What's going on? And a lot of times it's nice to stand and sit and rotate. So we're giving you that flexibility there. We even have a built-in work surface here that can rotate or lock into place. You can set your laptop, your extra samples, or anything else you might need while you're running your system. Of course, a system like this also needs powerful software to control it. So we have Sasquatch software, SQS for short. We have full experiment design capability that you can use offline to really optimize your experiment and the fluorophores you've selected for regular compensation experiments or for spectral. A lot of nice features in here also include system health, so we can notify you when any part of the system needs attention, and we can even monitor that remotely through Propel Cloud services if you choose to sign up for that. There's so many options. I could talk a long time on this, but I know we want to get to the technical detail. If you have more questions, please go to our website and check out all the technical details, as well as our launch if you'd like to view that. I'm going to pass it over now to Mike Kistner and Nate Trujillo. Mike Kistner at Columbia University has the first Bigfoot system installed, and Dr. Nate Trujillo is our application scientist here. They're going to give you a lot of detail on all the aspects of Bigfoot that allows the accurate high throughput plate sorting.
Thanks, Angie. Let me just share my screen here. Share and then sound. All right. So thanks a lot for the introduction, and I want to thank both MetroFlow and Propel Labs for the opportunity to speak here. So today, Nate Trujillo and I are going to be talking about the innovative and powerful plate deposition capabilities of the Bigfoot sorter. I'll be talking about the hardware features that have really impressed me as an end user, and Nate will be talking about some very compelling data he's generated using the HRP colorimetric method, demonstrating these capabilities. We've also put together a white paper with the content we will show you here today, and you can get that on the Propel Labs website, and there's a lot more detail on the white paper. I'll just get my laser pointer. So I just want to give you a little bit of an introduction to myself and my institution. I manage the flow cytometry shared resource that's part of the Columbia Stem Cell Initiative, or CSCI, at Columbia University. The CSCI is directed by Emmanuel Passaguet, and it's Columbia Stem Cell Program. We currently have 59 labs in the program across all 3 Columbia campuses, including 4 labs in our dedicated space at the Columbia University Irving Medical Center campus. CSCI fosters collaboration and community between stem cell researchers across Columbia. We host a variety of programmatic events like seminars, work in progress, retreats, and happy hours. And we also provide opportunities for funding for member labs. And in addition, we have 2 core facilities. We have CSCI flow cytometry, and we have the stem cell core facility, which is directed by Barbara Corneo. And this core facility provides a variety of services, including iPS cell generation, especially from patient samples, stem cell differentiations, and CRISPR-Cas9 genome editing. I also wanted to give you a brief introduction to the CSCI flow cytometry core facility. So we're a shared resource facility that provides access to flow cytometry instrumentation and services to labs across the Columbia campuses. We currently maintain 6 flow cytometers, 2 ARIA cell sorters, a BioRed S3 cell sorter, a Z5 analyzer, Faxel ESTA, and we recently added an ImageStream X Mark II cytometer to our offerings to further expand the core's technology. We have 2 staff members, myself and Dan Trost, we both manage the day-to-day operations of the core facility. And we also just added a new instrument to our family of cytometers, which is the Bigfoot, which Angie mentioned. And we've had the privilege to be part of the Bigfoot beta team, which is a team of 16 cytometry professionals across the United States and Europe, which was assembled by Propel Labs in early 2018. The team has provided feedback to the Propel group throughout the development process of the Bigfoot Cell Sorter. And it's been an amazing experience for us to be part of this group. And Propel clearly took a great deal of our feedback into consideration. And the instrument is truly a product of this collaboration. So as part of this collaboration, the Bigfoot beta team collaboration, Propel planned to install a few Bigfoot beta units across the country earlier this year. And we were fortunate to receive the first Bigfoots, serial number 1, back in early March. However, we shortly shut down 2 weeks later due to the COVID situation. So we unfortunately had very limited time on the instrument, just about 2 weeks. Regardless, I was extremely impressed from our time with the instrument during these 2 weeks. And a lot of what I'm going to talk about today is directly based on the testing we've had the opportunity to do ourselves. So the purpose of today's talk is to highlight some of the key features and performance of Bigfoot's plate sorting capabilities. And while most of you are probably familiar with this feature from your own experiences on other sorters, I wanted to briefly review it before discussing Bigfoot. So deposition of down to single cells directly into either PCR or tissue culture plates is a feature on most cell sorters available today and is commonly used for genomics, single cell cloning, or other plate-based assays, for example. So in order to accomplish this, a mechanical device or arm on which a plate is installed can be moved to various positions underneath the sorting hardware. And cells are deposited into the plate through a single dedicated side stream. And after the required numbers of cells are deposited into a target well, for example, A1 in this position, the plate will move to another well it will move another well under the side stream, so cells can be sorted into that well. And sorting proceeds so on and so forth until all of the required wells are sorted. So while sophisticated and impactful plate sorting experiments have been performed on available systems, there are some limitations. Firstly, deposition accuracy is critical, so the instruments need to be designed with very accurate plate positioning systems. Imprecise movement of the plate, especially when sorting into 384 well plates, which have a very small target area, can result in failure of alignment of the sort stream with the well and then possibly an empty well, which can be a severe issue if the downstream experiment is expensive, like single cell sequencing, for example. Secondly, speed is an issue because when the plate's moving from A1 to A2 or between wells, the sort is paused because the well needs to be underneath the sort stream in order to capture the cell. So during this process, the sort is paused. So cells are going to be lost while the plate is moving. So the faster that the plate moves, the better the yield will be. So lastly, one potential issue is that only one side stream is used. So only one population can be sorted at any particular time. So if more than one population needs to be sorted, they have to be done sequentially. And this is a particular issue in our core facility because a lot of our users need to sort multiple populations into plates. So it can take a lot of time and you can waste sample because you have to go through one population at a time. So I wanted to talk more specifically about introduce you to how the Bigfoot accomplishes plate sorting. So because the engineers at Propel were able to design the system from the ground up, they were able to tackle these problems through very thoughtful and careful instrument design decisions. And the main questions they tried to answer in designing the instrument was how can speed and accuracy be maximized for this process? So the answer they came up with to maximize the speed of a plate sort was essentially to multiplex the process by using multiple side streams instead of just one. So therefore, Bigfoot is capable of accurate 4-way 96 well plate sorting and 8-way 384 well plate sorting. So for example, for 96 well plate sorting, instead of employing just one stream, Bigfoot uses 4 streams at once. So rather than having to move the plate after every sort, the instrument only needs to move the plate after every 4 sorts. So the instrument is able to sort an entire 96 well plate by making only 2 passes across the plate. So here I made a very simple diagram trying to illustrate this. So this is a 96 well plate, and here 4 side streams are being generated to sort into A1, B1, E1, and F1 simultaneously. So what will happen is the instrument will make a first pass across the plate by sorting into rows A, B, E, and F. And after it's done sorting all the wells in those rows, it will move to C1, D1, G, and H and sort these rows. So therefore it could do an entire plate in 2 passes. Similarly, with 384 well plates, 8 side streams are used, which allows the completion of an entire plate in 2 passes as well. So this is incredibly fast, and the instrument can complete a 384 well plate in just seconds, 10 seconds. Additionally, which is also very impressive, is that the instrument can sort multiple populations simultaneously onto plates because it can make 4 and 8 side streams for 96 and 384 well plates respectively. So this is not something that's possible on other instruments. It can really help yield when sorting rare cells or samples precious and you can't really go through multiple rounds of sorting. So to illustrate just how the process works, I wanted to show you a video of a 4-way sort using a 96 well plate. So let's see. Here we go.
So here, We have an example of sorting 5 droplets onto the lid of a 96 well plate. For experiments where speed is a necessity, accurate 4-way sorting is critical. For the purposes of this video, we have overridden the sort door so you can watch as the sort output completes the entire sort in only 2 passes.
So here you kind of have an idea of how fast it is and really what it looks like when it's sorting a 4-way sort for a 96 well plate. So to go a little bit deeper into how this is possible, I wanted to discuss 3 of the subsystems that, for me as an end user, are standout systems and really are able to accomplish this and show a lot of innovation in design. So the deflection system is one that really allows this kind of sorting to be possible. The sort output mechanism and the electronic system. So I'll discuss each of those in more detail, but before we move forward, I wanted to just give you an overview of what the sort area looks like. So on the right in this diagram, you could see the multi-tube sample loader, which Angie talked about. And on the left is the sort output area. So you can see the deflection plates, which I'll talk about in more detail, the waste catcher, and then the sort output mechanism on which a 384 well plate is installed. I also want to show you the streams imaging system. So this is a sophisticated system using LED illumination, mirrors, and camera system that can image the side streams for automated setup and monitoring. So that is a very important part of sort setup in general, because the instrument will automatically align the streams to targets and thus to tubes or the plate. So I want to start talking about the deflection system. So when designing the Bigfoot Cell Sorter, the engineers at Propel realized that in order to generate multiple side streams to perform this multi-way plate sorting that we've seen, they needed to make sure that the hardware was designed to maximize side stream stability, especially when creating 8 side streams. So one issue that the engineers needed to overcome is that charge droplets can interact through attraction or repulsion, and this can lead to instability that results in fanning and potentially severe issues with the sort and the collected fraction. And these issues become more and more relevant as more side streams are generated and more charge droplets occupy a smaller space or in closer proximity. And it was clear that in order to generate the number of side streams required, the amount of charge on droplets needed to be minimized as much as possible. And the engineers were able to do this while still maintaining the required deflection angles for 384 well plates and for 6 well sorting for tubes by designing an exceptionally long deflection path. And this allows droplets to be gently pulled out of the stream and directed to their collection device with minimal charge magnitude. Additionally, increasing the geometry of the path length also permits more side streams to be generated simultaneously, because the further away the collection device is from the point where the streams are generated, the more divergence in the side stream angle, and this allows sufficient spacing between the streams to accommodate collection tubes of typical diameters that we usually use, or multiple wells of a plate. Another aspect of the deflection system is the custom designed deflection plates, which also function in addition to the path length to maximize side stream stability. So these plates have unique geometry and size that are designed to permit adequate deflection while minimizing excessive droplet charge. Additionally, they're also very easy to clean, which is critical for stable side stream formation, because salt buildup, for example, on the plates can disrupt the field they generate and cause deflection issues and unstable side streams. So to illustrate this, how stable the side streams are in more detail, we've prepared a video of an 8-stream test pattern. And this is the test pattern that is used to set up a 384 well plate, 8-way sorting for a 384 well plate. And the images in this video are captured with the side stream imaging system. And additionally, while you watch this video, I want you to notice that the software is going to automatically adjust the streams to calibrated targets, which you'll see at the bottom of the image. And this brings up another point. A lot of the setup steps for plate sorting and tube sorting are automated on Bigfoot, including side stream setup. So the instrument will set up the side streams for a particular target, so you don't have to do that manually like on other systems. And this process works very robustly based on what I've seen with our testing. We found that plate alignment only needs very slight adjustments from the predefined targets from day to day. So let's take a look at this. So you can see here are the 4 left side streams, 4 right side streams, and then the center stream. And you're going to see here, the software is moving the streams, adjusting the deflection to line up with the targets. And you'll see it's doing all the streams. And after this process, we would typically test the alignment by sorting onto the lid of the plate to make sure that it's valid. And only very small adjustments seem to be necessary from day to day. So moving on to the sort output mechanism. So this is very impressive to me and was clearly designed with a great deal of thought. And as you saw from the video demonstrating sorting of a 96 well plate, the movement is very fast and the precision is very high. So this is part of the instrument that really makes this this capabilities of plate sorting possible. So the instrument was designed with a few specs, target specs, and for accuracy, it was designed to be able to deposit droplets within 0.175 millimeters or 10% of the surface area of a well of a 1536 well plate. And this is a very small target. Additionally, in terms of speed, it was designed to be able to be capable of traveling the entire length and width of a well plate in half a second with minimal lag during transition between wells. And as I mentioned, this is very important because any lag in transition will result in lost cells, target cells, and a lower yield of the sort. And this is all accomplished through sophisticated hardware, including a custom designed linear actuator system, fine pitch lead screws, a high torque motor, and an intelligent spatial sensing system to minimize any variability in positioning between the wells. So this is clearly something that was very carefully designed to optimize plate sorting, the precision required for plate sorting. So I want to show you another video. This is what I kind of consider to be the pinnacle of the capabilities. And this is the high-speed 8-way 384 well plate deposition, which clearly takes advantage of the stability of deflection and the robustness of the sort output mechanism in terms of accuracy and speed. So take a look at this here.
The Bigfoot Cell Sorter is capable of doing 8-way sorting into 384 well plates. In this video, we're sorting 1 droplet into each position onto the lid of a 384 well plate at unprecedented speed. Just like the 96 well plate, we can finish the entire sort in only 2 passes. With a 384 well plate, this only takes Bigfoot 10 seconds.
So you can see the spacing between the puddles is really, really consistent. So we're really excited to do more testing with this. Once we get back in the lab, we're going to try to test this with various different cell types and cell sizes and further test the accuracy. And Nate will talk about some of the future directions as well after his talk. So finally, I just wanted to mention a few points about the electronics. The electronics are an extremely powerful part of the machine and were designed from the ground up for speed and performance, and really highlight and really contribute to Bigfoot's plate sorting capabilities. So the first thing I want to mention is that all operations are executed in real-time hardware control as opposed to software. So the entire list of operations, everything that the instrument has to do to move the plate, for example, is programmed ahead of the sort and downloaded to the instrument. So this eliminates the need for workstation and operating system to do any work, which will minimize lags. And additionally, the electronics were designed with what's called a dynamic window extension that facilitates event processing of over 100,000 per second without any electronic aborts. So compared to a fixed window extension, which cannot independently classify events that occur within the same analysis window, a dynamic window extension can snap to pulses. So pulses that occur close together can be processed by the hardware independently and included in the data set. However, it's important to keep in mind that doublet events can still occur if pulses overlap significantly. So this has to be gated out. Doublets have to be gated out as usual, like they are on any instrument. The dynamic window extension really is to minimize electronic aborts. Also, the drop charge delay measurement is calculated from the center of the pulse or droplet cycle rather than the edges of the droplet. And this strategy for calculating the drop charge delay has the potential to minimize the impact of particle size on the drop charge delay. And this is something we really want to test thoroughly once we're back in the lab. We've done a lot of testing on our other instruments, so we're looking forward to adopting that to the Bigfoot. And finally, something that's very convenient is that the hardware was designed to save index sorting data automatically. So you don't have to turn on this feature. And this can be very convenient if this is something you need later on and you didn't realize to collect when you are sorting. So that's all I'm going to talk about today. And I just want to summarize briefly before I hand it over to Nate how impressed I've been with all the innovations that Propel has thoughtfully considered when designing the Bigfoot from all the testing we've done. And I'm really looking forward to continuing to explore the instrument once we're back in the lab. I also want to truly thank the Propel team for taking all of the 16 members of the beta feedback group's consideration so seriously. A lot of our suggestions were clearly integrated into the machine, and it's been great to be part of that process. And I really look forward to exploring the technology further with all the challenging projects that come through our core once we're back in the lab. So now I'm going to turn it over to Nate, who's going to talk about some very compelling data he's generated to further demonstrate the Bigfoot's capabilities, plate sorting capabilities.
Thank you, Mike, for a really great overview of the sorting capabilities of the instrument. That was really nice. So I'm going to be talking some more about the experimentation we ran here at Propel to highlight some of Bigfoot's high-speed plate sorting capabilities. Originally, these tests, like Mike mentioned, were planned to be run on Mike's beta Bigfoot system, but the COVID situation kind of changed those plans. So fortunately enough, Mike would continue to collaborate with us and provided amazing insight and ideas. But before I get into that, I want to circle back a bit and reemphasize a few of the key points that Mike touched on regarding the importance of single cell sorting. So high-speed sorting into multi-well plates has been known to be kind of the standard for cell purification for a wide variety of downstream applications and experiments. And as you can see in the short list below, plate sorting can often be seen as one of the first steps in a study that leads your experimentation into other realms of cellular and molecular research. This method, though, often comes with some limitations and challenges that researchers often face with current instrumentation. These include some time restrictions to run multiple plates, and then the precise alignment of the side streams to the media target wells, especially when using things like 384 well plates or 1536 well plates, because the targets for those wells are very small. And you also have to take into consideration differences between different plate types. So a difference between a 384 well tissue culture plate and a 384 well PCR plate. The geometry of the wells are different, and the sizes are different for those wells. You have to take that into consideration when you're aligning your streams to those wells. But this leads us into the method that we're going to be talking about today, which is the HRP deposition method, in order to kind of help us facilitate with some of these challenges. So HRP, or horseradish peroxidase, was proposed as an alternative for immunoassays back in the 1970s, and it's found in the roots of horseradish, and it catalyzes the oxidation of various organic substrates by the use of hydrogen peroxide. By itself, the HRP enzyme doesn't have much value in biological applications, but when it's used in the presence of a substrate and an oxidizing agent like hydrogen peroxide, the reaction produces a significant color change that can be seen visually or can be taken using spectrophotometric methods, where you can measure the relative intensity of the color change itself. So for those of you that are really familiar with ELISAs, TMB is a common substrate used in ELISAs and is a reagent for detecting peroxidase. The high sensitivity substrate solution that we use often contains not only TMB, but hydrogen peroxide and other stabilizing agents. And it's colorless, or a faint yellow when it's in its unreacted form. When oxidized by HRP, will produce a vivid blue color almost instantly. So Rodrigues and Monard back in 2016 decided to take advantage of this mechanism and reaction between HRP and TMB and utilize it for sorting as a rapid method for verifying single cell plate deposition. And depending on the concentration of HRP you're using, the volume of HRP needed to produce the color response is very minuscule. So it's fantastic for flow cytometry and sorting, since the carrier droplets for particles and cells are so small. So just to quickly go over the materials used in these two experiments that you're going to be seeing in the upcoming slides. We use lyophilized salt-free HRP, which we reconstitute in HBFS to a working solution of 5 milligrams per ml. And we wanted a concentration to be on the higher end so that the color change would be very vivid for visual inspection or for photographs, which is what we were going to be taking. In addition, we added a small volume of 4.5 micron polybead carboxylate microspheres to the HRP solution to use as our sample on the Bigfoot. And again, these were kind of methods that were adapted from Rodrigues and Monard back in 2016. And then we added TMB high sensitivity substrate solution to the 96 and 384 well plates at 50 microliters of volume to the 96 well plates and 10 microliters of volume to the 384 well plates. In both of these experiments, we were using PCR amplification plates because of the circular wells and the conical bottoms, which allows us to utilize smaller volumes. And then using these plates kind of help closely mimic the types of experiments that you would potentially see brought to the Bigfoot Cell Sorter or high throughput plate sorting. And then we also wanted to ensure that we highlighted the speed and the repeatability of the plate sorting on the Bigfoot. So we ran 5 of each plate type in order to see how fast can we run multiple plates on the system. So the setup for this experiment was adapted from, again, Rodrigues and Monard. And we divided it into 2 parts. So for part A, we wanted to demonstrate multi-droplet deposition into 96 well PCR plates using the 4-way sorting capabilities on Bigfoot. And then for part B, we wanted to demonstrate single droplet deposition into 384 well plates using 8-way plate sorting capabilities on Bigfoot. And using the multi-way sorting, we wanted to accomplish the similar results of the manuscript while maximizing the speed and maintaining the same accuracy. So for part A, using 5 plates, 96 well PCR plates containing 50 microliters of the TMB solution, we sorted 4 target events into row A, 3 sorted target events into row B, 2 into row C, and then 1 into rows D, F, G, and H. And then in the middle row, row E, we excluded this from the sort logic. That way we would have a control reference row with the unreacted TMB solution. And then for part B, using 5 384 well PCR plates containing 10 microliters of the TMB solution, we wanted to sort just 1 sorted target event into alternating odd rows while excluding the even rows from the sort logic. And again, this was so that we could have rows of unreacted TMB as a control reference after the reaction. And then all sorts were performed at 150 events per second using the single cell sort mode. And in this mode, 1 drop is sorted if both the target drop and the adjacent quarters of the neighboring droplets do not contain any other events in them. And then if they do, the droplet is aborted until another droplet fits that criteria. So before I get into the results, I wanted to go over kind of the workflow of setting up the plate sort on Bigfoot. And the reason why I wanted to highlight this is because the amount of time and effort it takes to set up a high throughput experiment on a sorter can be really challenging and plays a significant role when it comes to scheduling and planning out an entire study. So one of the great things we've done on Bigfoot is to automate many of these processes with sort setup. That way we can minimize the time that you spend doing this and maximize what you really want to be doing on the instrument, which is running your sample and sorting. So starting at the beginning, we have the instrument startup, which takes less than 2 to 3 minutes for fluidic startup to prime the sheath, debubble the nozzle tip, and power on all the lasers. And again, this is all automated just by 1 click in the software. Once the system has been on for a bit and it's given a little bit of time to settle in, we can now move to the automated QC alignment and verification that Angie mentioned earlier. And this process is a 2-part method where we use our onboard calibration bead mix. And during part 1, we can gate on a single peak, which fluoresces in all channels. And we do a fine adjustment of the stream at the laser interrogation point in order to optimize its position, in addition to auto voltage adjustment and auto laser delay adjustment. And then for part 2, we gate on a 2 bead mix with the single peak bead that's our bright and a blank bead in order to calculate a separation index on all fluorescent channels. And this allows us to look at all the detectors and verify that we have adequate separation between the negatives and a bright signal on a log scale. And the second part also gives you some starting voltages if you choose to utilize them, kind of as a baseline for multi-color compensation experiments or for spectral and mixing experiments. Going on to setting up the droplets, streams, drop delay, these are all automated systems in which a self-run user can perform this on their own without the need for in-depth technical knowledge of the system. We enable drop drive at the beginning of QC and do automatic calibration of the frequency, amplitude, charge phase, and defanning just at the click of a mouse. Drop delay is calculated using the third bead that Angie mentioned in our onboard bead mix. And in this process, we deflect the waste stream to the right and then deflect the sorted stream to the left where it is caught in a dedicated port on the waste catcher. And it runs basically through a mini cytometer in the back of the instrument and does a sweep of various drop delay values in order to give you the correct calculation for your drop delay. And you can kind of see that in the photo on the bottom where we get this nice peak when you're starting to reach the correct drop delay calculation. From there, we do automatic adjustment for the stream deflection, as you saw in the video that Mike had showed earlier. And this is just using a software-generated test pattern in order to do individual adjustment of each side stream to the calibrated targets that you have using the stream's imaging system. And all of this instrument hardware setup is kind of part of, you know, a lot of different steps that you have to do in order to get ready for a sort. But we've included this all into this automated process where a lot of these, you don't have to spend a whole lot of time doing that. And then you can get straight to your experimental setup, which is where you're going to be setting up your plots, your regions, your sort gates, adjusting the voltages and setting your sort logic. And then you are ready to sort. Now we'll get into kind of the results from the methods that we were doing before for this experimentation. So for part A, which is the multi-droplet deposition onto a 96-well PCR plate, in the representative image on the left, you can see that after 30 minutes of incubation in the dark post-sort, there's an obvious gradient of dark blue to pale blue as we move down from row A to row D. And then 4 sorted droplets to 1 sorted droplet. And then as a reminder, the last 3 rows contain only 1 sorted droplet. And the middle row contains just the unreacted TMB because we excluded this from the sort logic. As I mentioned before, this is just a representative image of just 1 plate out of 5 that all produce similar results. So we got the color changes that we would have expected based on the methods that were adapted from the Monard paper. But we really wanted to highlight here was the speed at which we can perform the sort. So we recorded the elapsed sort time to complete a full 96-well plate from start to finish using 4-way sorting. This is the time it takes from when the user clicks the sort button in the SQ software till the end of the sort when the plate is back in its origin position for user retrieval. And as you can see in the table on the right, the elapsed sort time for all 5 plates were very similar. And you get an average sort time for each plate of 7.89 seconds, which is really fast. And keep in mind that these plates were run sequentially without the need to recalibrate or rerun a drop delay or do any other recalibration of the instrument. Then moving on to part B, which is the single droplet deposition, you can see that with the representative image on the left, after 15 minutes of incubation, the color change of reaction is very evident with the deep blue coloring in the odd rows and the unreacted TMB substrate in the even rows. And as expected, these results are very similar to the manuscript, but what we wanted to take further is highlight the speed at which we can accomplish this using 8-way sorting into 384-well plates. And in the table on the right, you can see the elapsed sort time of a single droplet, 8-way deposition into each of the 5 384-well plates. And again, this timing from the start, this is the timing from the start of the sort to when the sort was completed and back in its home position for retrieval from sort output. And the average sort time across all 5 plates was just 10.42 seconds, with the plates all being run sequentially without interruption. So you can imagine with the speed and ease of this on the Bigfoot Cell Sorter, you can imagine how this would streamline your workflow and allow you to move on more quickly to other downstream applications once you have multiple plates sorted and you're not spending so much time doing the actual sorting itself because we've kind of made the speed and accuracy so great on the Bigfoot. So I just wanted to conclude here just a few of the key points just from Mike and then from the experimental studies that I just talked about. The hardware innovations incorporated into Bigfoot allow it to overcome many of the challenges that users often face on other platforms. As Mike pointed out earlier, the path length and geometry of the deflection system permits for up to 8 sort streams that are highly stable from one experiment to the next and allows for flexibility in multi-way plate sorting applications. The speed of the movement and accuracy of the sort output platform while sorting reduces the lag time between well-to-well movements, thereby maximizing the yield and reducing the amount of sample used to complete one plate. And finally, the electronics of the system with real-time hardware control of operations frees up the software and the strain on the workstation from having to continuously respond to and update changes in status during the sort. These high throughput plate deposition experiments clearly highlight the plate sorting performance of the Bigfoot, which truly sets it apart from other platforms in terms of speed of accuracy of multi-way sorting, repeatability without the need for users to recalibrate their side streams or rerun a drop delay between plates. So this makes the workflow just really streamlines it a whole lot better for you and saves you a whole lot of time. So as Mike mentioned earlier, there's some future work and plans that we have in place for kind of continuing on with some of this experimentation. A lot of this is going to be planned to be performed this summer on Mike's beta instrument that's still at the CSCI. We plan on doing some HRP-TMB methods repeated with some smaller volumes down to 2 microliters on 384 well plates. I'm going to be utilizing 8-way, 4-way, and straight down sorting. That way we can compare the speeds of all 3. We also want to do some custom sort positioning across 96 well and 384 well plates. That way you're not limited to sorting in just rows or columns, and you can pick any position across the plate that you want to sort into. And then sorting in multiple populations with varying concentrations. So we have the ability to have 4 side streams or 8 side streams, and we can designate 1 population to each of those streams, and then look at varying concentrations with all those. We also want to test whether or not the sorted droplet that we had deposited into the plate contained the target cell or particle of interest. So one thing we can do, I think users often do too, to verify this is to do fluorescence microscopy with a flat bottom plate. Or we can take a more thorough approach and do some genomics in order to look at that. And then finally, kind of as I mentioned, we can look at measurement of the color change using the same method, and then looking at a relative intensity of the color change using spectrophotometry. So before I move on to the Q&A, I wanted to quickly thank Mike for working with us and providing some excellent feedback as a beta Bigfoot user. And then I also wanted to recognize the Propel Labs team. So for those of you who aren't really familiar with us, we're based out of Fort Collins, Colorado, which is about an hour drive north of Denver. And our team consists of just under 40 very talented individuals, not only in Colorado, but across the US and in Europe. And if you're interested in learning more about us or about the Bigfoot Cell Sorter, feel free to visit our website below at www.propellabs.com. And with that, we'll be happy to take some of your questions. Angie will be moderating some of our questions today, and then joining us for the Q&A, we also have our President and CEO, Tidar Sadeh, our Director of R&D, Dan Fox, along with Mike and myself. And I want to say thank you to all the attendees that came today and joined us, and also thank you to MetroFlow for having us for this talk.
Thank you so much, Nate and Mike. It's wonderful to go into all that detail. And as Nate mentioned, we do have a white paper and more detail on our website if you missed something. I know we were answering lots of questions while we were going, but I saved a few for live. So let's start with one for Dan. Dan, does the accuracy suffer when multiple streams are used?
That's a great question. So you do get the best total accuracy by doing straight down sorting. Without any charge, without any deflection, it just takes some of the variables out of the equation. However, what we were able to do with the Bigfoot, with everything you saw in the presentation, was get it so that our accuracy was good enough to be able to know that we had to be able to hit a 384 well plate target by doing up to the 8-way deflection. So if you're doing a 1536 well plate, or if you were going into a really tiny amount of volume and a V bottom and you needed it more than average precision, we do support straight down sorting. And the way we support that is by having a dedicated position on the waste catcher. So we can move that and deflect the waste stream, and that's all done automatically by the system. So you don't have to try to reposition the waste catcher or do any manual adjustments to the system. It's all just in how you've configured your sort for what you're doing. But for most applications, and the 96 and 384 well plates, the accuracy is definitely good enough to be able to do it with the multi-way sorting.
Thanks, Dan. We also had a couple questions on the 10X adapter. I know it's not directly related to this, but there was questions on how does this work? And I'll just take a quick stab at that. The adapter just fits on top of the sort box, and we can sort 8 ways directly into that chip. There were some further questions about volume for those droplets, so I'm going to let Dan answer that.
So depending on your sort setup, from 70 micron to 100 micron tip, you'd be looking at 1 to 3 nanoliters per drop. is very close estimate that I always use. So it's really pretty simple, 1 to 3 nanoliters per drop, and that's the faster you're going, which is the smaller tips, so like a 70 micron tip at 60 psi would be about that 1 nanoliter, and doing a 100 micron tip at 30 psi at around 40 kilohertz would be more like 3 nanoliters per droplet.
Thanks, Dan. So we had one other question about plate sorting time and relative frequency of the population to be sorted. In fact, we had lots of questions about that. So of course, the plate sorting time is dependent on the frequency of the population, and it's also dependent on your event rate and your purity settings. So those all need to be taken into account for every specific application to really optimize the time, which of course is the limiting factor we all have. So we have been working on efficiency tools to show you what the optimum rate would be for the maximum efficiency for the sort percentage that you're sorting to help guide you in that area. But it really is a specific case by case and dependent on nozzle tip size and so many other things. So those are all the additional questions I had. We really appreciate all your time. We appreciate you, MetroFlow, for allowing this to happen, and our panelists and all the details. Are there any more questions before we sign off? Oh, I do have some more popping up. So we have a question, Dan, for you. Can you manually set sort frequency and have the instrument calculate appropriate deflections?
So we do have the system where it automatically sets up the stream for what would be your normal operation, and that's based on the tip size that you have selected. We do have the ability for you to adjust kind of what that top point is. For a 100 micron tip at 30 psi, you could adjust the frequency to say, no, I want it over here, and kind of have that be the configured point that it starts with. So there is flexibility for that. And then for the appropriate deflection, yes, that's a separate step in the overall setup. So once the stream stuff is set up back at the point where it was kind of taught to start looking, then when it gets to the deflection setup, it would still do all of that automatically.
Thanks, Dan. So another question we have is the long distance deflection path, does it have any effect on cell viability? And I've had a couple questions actually on viability. Generally, jet and air is more gentle on cells during the sorting process, and all the other features we put in the system really help support good viability. I'll let Dan cover that in a little bit more.
Yeah, and certainly the temperature control that comes standard for the input and the output helps a lot with viability as well. The deflection distance doesn't have any impact that we've ever been able to measure or detect in looking from very short to very long. The acceleration profile during flight there's almost no changes going on. And the size of the droplet, it's not evaporating, it's not changing temperature considerably. We're talking about, I think it's about 15, 20 milliseconds total flight from top to bottom. And it doesn't really matter how far it goes during that for viability.
Okay, any other questions? So this is a good question actually. Fanning is often an issue in sorting, and this would ruin multi-well plates. So how is this problem avoided?
There's a lot that goes into the defanning, and a mix of that has come from some instrument issues over time. There's been a challenge with trying to get enough separation so droplets that have a very positive and a very negative next to each other can attract to each other and merge and cause additional side streams. And we've avoided that by being able to really control the distance that we have and do optimal charging at a much lower charge than other systems. So we don't have that problem. We do also have a very good imaging system set up to try to keep the fanning as tight as possible from the initial settings. And we've built in some advanced algorithms that are help looking at the different patterns that are coming through, because there's electrical effects, there's physical effects, there's trajectory effects. There's a lot of things that all come in, and we've put a lot more into the overall system than we ever have before or have ever seen anybody do before. So we have, in my opinion, the tightest streams over the widest flexibility on sorting output options.
Thanks, Dan. So a couple more questions that we have. One, are the times demonstrated for sort on a 70 PSI pressure? Somebody's asking, sorry. So I'll let Nate touch base on that since he did most of the work shown.
So these were all done just using 100 micron nozzle. That will probably be included in our future work. I know Mike has all of the different tip sizes that we have included on the instrument, and I know that he's eager to try a lot of different experimentation with the different tip sizes, one being the 70 micron nozzle. So that'll be part of the future work that we do.
Thanks, Nate. And the last question I have here is, you have up to 60 different detection channels. Do you have any pre-made panels that fill all of those? What a great question. We actually have all these channels, first of all, to enable the spectral capability, and then second of all, to give you the flexibility to adapt whatever experiments you have or want to expand to the next level. Currently, we don't have any pre-made panels. We do have a white paper on a 15-color spectral sort on our website if you'd like to take a look at that. But we have designed the instrument not to limit your science. We want you to take that to the next level. Okay, we thank you all. I think we're at the time, so we thank you all so much for your attention, and thank you so much, MetroFlow, for allowing us the presentation. Tina, did you have anything to add?
Yes, thank you. Thank you, Angie, very much, and thank you, Mike and Nate. We really appreciate your presentations. And we have about 60 attendees left on the call. We are not sharing your email addresses with the corporate sponsors for these seminars. So if you do want more information, please reach out via whatever the Propel website and whatever other contact information was provided during the slides to get some additional information. So with that, I will go ahead and wrap up. Thank you all for attending. Everyone stay safe and healthy, and we look forward to seeing you virtually on our next event. Thank you.